Hi everyone, my name is Mary Sila, and today I will be presenting on my topic of protecting the gut microbiome from carbapenem-mediated damage. Throughout my presentation, I will be referring to Sheila Connolly's oral metallo-beta-lactamase protects the gut microbiome from carbapenem-mediated damage and reduces propagation of antibiotic resistance in pigs. The flow of my presentation will begin with background on what carbapenems are and their importance, followed by the methodology of the article. Then I'll go into the results and the observations made, followed by a discussion. Let's get started and hope you guys enjoy. So first off, I want to begin by discussing what carbapenems are and what the importance of them are. Carbapenems are a class of broad-spectrum antibiotics that are highly effective against many bacteria, including those that are resistant to other antibiotics. They are considered one of the last resort antibiotics, often used when other antibiotics fail to treat serious infections. Carbapenems are beta-lactam antibiotics, similar in structure to penicillin. However, they possess a unique structure that makes them highly resistant to degradation by most beta-lactamases, which are enzymes produced by bacteria that can break down beta-lactam antibiotics. Carbapenems work by inhibiting bacterial cell wall synthesis, leading to bacterial cell death. Common examples of carbapenems include imipenem, meropenem, and eripenem, which we will be discussing further throughout this presentation. Due to their potency and broad spectrum of activity, carbapenems are primarily preserved for treating severe infections caused by multi-drug resistant bacteria, such as certain strains of Enterobacterales, Pseudomonas aeruginosa, and Acinetobacter baumannii. They are often administered intravenously in hospital settings and are used very cautiously to minimize the risk of promoting antibiotic resistance. However, carbapenems are not infallible against bacterial resistance. In fact, they can undergo resistance through mutational events or through gene acquisitions via horizontal gene transfer. There are 5 major carbapenemase genes that are found, which include KPC, BIM, M, oxytype, and NDM. We will be going further into the oxytype carbapenemases within this research. CPOs are carbapenemase producing organisms that are resistant to carbapenems. This is a major threat for HAIs, aka health-associated infections, because they target immunocompromised patients within hospitals. These infections are CREs, carbapenemase producing Enterobacterales, CRPAs, carbapenemase resistant Pseudomonas aeruginosa, and CRABs, carbapenemase resistant Acinetobacter baumannii. This graphic from the CDC shows that between 2017 and 2022, over 95,000 CREs were isolated and tested, with over 30% having at least one carbapenemase resistant gene. So I think that this really shows how important it is that we focus on carbapenemase producing organisms or carbapenemase resistant organisms, just because it is on such an incline. So now, what is the medical importance? It is no secret that antibiotics have been misused in their applications, including within hospital and outpatient use by physicians through unnecessary or incorrect prescribing, by patients through incorrect course dosing and durations, and on large-scale agriculture. The danger in this is that CPOs are resistant to nearly all beta-lactams available on the market. This leaves only older antibiotics like polymyxins and fosomycins to treat these infections. This is an outcome that should be avoided, it needs to be avoided, because antibiotics like polymyxins cause kidney toxicity in almost 40% of patients treated with it, making it a very dangerous antibiotic to use. In regards to the human microbiome, there is a connection between carbapenem use and the gut microbiome, since it has been found that gut dysbiosis promotes the overgrowth of antibiotic-resistant pathogens like CREs and CRPAs that we have discussed. Now, what are the implications behind this? Immunocompromised individuals are highly susceptible and in danger of these pathogens and have limited treatment options. In addition, antibiotic resistance throughout the world increases as these carbapenemases spread. This is an urgent global public health problem. In fact, in 2016, the CDC developed a program called the Antibiotic Resistance Laboratory Network, ARLN for short, that aims at surveillance of this problem. The way I actually got interested in what I'll be presenting today is that I actually worked through this program at the Florida Department of Health, and I actually specifically work with carbapenemase producing organisms, and I was very interested in seeing what the connection between all of this antibiotic uses, its resistance, and the effect on the microbiome. Now we're going to discuss what has been done within this field. In 2019, an orally administered beta-lactamase was administered to see if it could provide antibiotic inactivation within the GI tract to protect the gut microbiome and to reduce antibiotic resistance. Ribaxamase, aka SYN004, was shown to degrade ceftriaxone in the GI tract and preserve the gut microbiome with patients suffering from Clostridium difficile, aka C. diff. It was found that SYN004 degrades penicillin in addition to cephalosporin beta-lactams, however, not carbapenems. Now the question becomes if another oral beta-lactamase could potentially degrade these carbapenems, which leads us to our research question. Does SYN006 have the potential to protect the gut microbiome and reduce the emergence of carbapenem-resistant organisms? Now I will continue on in speaking about the methodology of this research. Beginning with the test article, SYN006 is a formulated P2A. P2A is a novel beta-lactamase isolated from the Bacillus cereus and demonstrated to preserve colonization resistance in mice treated with antibiotic beta-lactamase inhibitor combination piperacillin tazobactam. SYN006 is formulated for oral delivery with a cellulose isolation layer to protect the enzyme from exposure to low pH during digestion. Please note that SYN006 will be the mechanism by which researchers are hoping to mitigate gut dysbiosis and lower antibiotic resistance. Within this study, 16 2-month healthy pigs with no previous antibiotic exposure from the Sus Scofford domestic Yorkshire cross were used as the animal model. Please note that pigs were chosen as the animal model because pig and human GI tracts share similar anatomy, immune function, and microbiota composition. In fact, 90% of the microbiota functional pathways in the human microbiota are represented in pigs. These pigs were then split into 2 cohorts. Group 1 received ermapenem, which is a carbapenem referred to earlier, and within this presentation will be abbreviated as ERT. Group 1 received ERT at 30 milligrams per kilogram intravenously once daily for 4 days. Group 2 also receives the same ERT, but with SYN006 at 50 milligrams 4 times a day for 21 doses, beginning the day before antibiotic administration. SYN006 was administered through a capsule and apple juice injection to ensure that the pig's stomach pH remained below 5.5 to prevent dissolution of the coated capsules and premature P2A release and inactivation before reaching the small intestine. ERT was administered through IV to animals under sedation at the same time daily. In terms of collection, blood and fecal samples were collected. Blood was collected on day 3 of antibiotic treatment after 1, 2, and 3 hours of antibiotic administration. Fecal samples were collected 2 times a day, beginning the day before any treatment was given and until the final day SYN-006 dose was given. In addition, the pig serum was analyzed for ERT using liquid chromatography turbo ion spray tandem mass spectrometry method following protein precipitation extraction. Statistical analysis were then performed. In terms of sequencing, total GNA was isolated from the fecal specimen using a QIAGEN kit and then library prepped using Illumina. Samples were then sequenced using the Illumina HiSeq with 225 million base pair reads per sample. These reads per sample were consistent with the equal read depth. These unassembled whole genome shotgun metagenomic sequence reads were analyzed to discover the bacterial identification to species, subspecies, and if possible, strain level, as well as the microorganism relative abundance using the distance measure, stacked bar graphs, and heat maps based on relative abundance of microorganisms in each sample. And finally, analysis of the bacterial sequence data included the alpha diversity and principal coordination analysis. And now we get to the meat of the presentation, the results. In terms of the discussion of results, it will be split into 3 sections. The first being that SYN-006 did not affect systemic ERT levels. Result 2, that SYN-006 mitigated ERT-mediated microbiome damage. And result 3 of SYN-006 reduces antibiotic resistance. So beginning with this first observation, it was identified that SYN-006 did not affect systemic ERT levels, aka the SYN-006 did not alter the serum antibiotics. To come to this conclusion, researchers treated one pig cohort intravenously with ERT 1 times daily for 4 days in a row. The other pig cohort also received this treatment, but they received the SYN-006 orally 4 times a day, beginning the day before the ERT treatment began. The pig's systemic antibiotic levels were evaluated to ensure that the SYN-006 was not affecting the uptake of ERT within the serum, meaning that it would not be affecting the concentration of antibiotic within the pig body. This is important within the experiment because the antibiotic must be able to treat whatever infection it is given for. Therefore, SYN-006 has to not interfere with the drug and bacterial interactions. This is an important consideration because maintaining correct serum levels optimizes the drug dosage regimens. So within this first observation from the result, researchers found that ERT levels were not statistically different between the 2 cohorts at any point in time, as shown in Figure 1. This blue-gray bar are the ERT-only cohorts, and this white bar is the ERT and SYN-006 cohort. So as you can see, the SYN levels did not alter the serum antibiotic levels in pigs between hour 1, 2, and 3, and there was no statistical difference found. Now we'll move on to observation 2, which was that the SYN-006 mitigated ERT-mediated microbiome damage. And the importance of this was that there was partial microbiome protection. So within this observation, the relative abundance of bacterial strains was determined to allow for microbiome composition comparisons. It was discovered that after antibiotic treatment, bacterial species declined by greater than 70% in the ERT microbiomes and by less than 50% in the ERT and SYN006 microbiomes. This means that there is a significant reduction in bacterial richness between the two species. The ERT only cohort resulted in a significantly lower alpha diversity ratio compared to the ERT SYN006 cohort. What was interesting, though, was that within the principal coordinate analysis, there was one isolate of the ERT SYN00 post sample that was closer to the ERT post sample than the pre-cluster. And this suggests that there is a partial microbiome protection. In terms of the pig microbiome, stacked graphs and heat maps were made as shown in this figure. So the importance of this top figure is that compared to the pre-treatment microbiomes, ERT resulted in a reduction or loss of specific bacterial strains and overgrowth of other bacteria in comparison to the ERT and SYN006 treatments, which has fewer microbiome alteration in composition. The ERT exposed microbiomes show a reduction in organisms including Bifidobacterium pseudolongum, Prevotella copri, Copricosis catus, and overgrowth of other bacteria, not observed in the ERT SYN06 microbiomes. I think something important to discuss that was also discussed in the discussion portion of the article is the dramatic overgrowth of an unclassified Sphaerotheca. The reason that this is important is because this is a genus that is found within members of the fecal microbiome of pigs, and they're inherently resistant to beta-lactam antibiotics, thus explaining the proliferation of the presence of ERT. Within this heat map below, I think it's very interesting because these black boxes display bacterial taxa diminished in the ERT cohort, however, are maintained in the ERT and SYN cohort. The white boxes show the bacterial taxa that proliferated with ERT and less so with the ERT SYN06. So essentially, this is showing the bacteria that were lost within antibiotic use versus the ones that overgrew with this antibiotic. In result 3, the SYN006 observation was that it reduces antibiotic resistance. And now I just wanted to discuss what is the importance. And the reason I left this blank is because I want you to take a second to consider what the background at the beginning of the lecture was. And the entire importance was that there is such growing antibiotic resistance throughout the globe that we really need to find ways to reduce this. So I find that this observation is very important. So it was found that by using the fecal DNA data, it was analyzed for the presence of antibiotic-resistant genes as a measure of population of antibiotic-resistant bacteria. So essentially what was done, the pre-treatment and post-ERT and SYN006 cluster remained together while the ERT resistomes diverged more than the pre-treatment from the pretreatment than the ERT-SYN resistomes. So that is shown here in red. This is the ERT-only resistomes, and you can see that it diverged from this black right here is the pretreatment, and the green and the blue are the ERT-SYN006 post and pretreatment. So as you can see here, there was a great divergence in this resistome. This shows that there was a reduction in the SYN antibiotic resistance gene propagation because there is no significant cluster differentiation and ERT-SYN, unlike this ERT-only cohort that drifted off. In addition, something interesting that was found was that 12 oxygenes were detected after ERT exposure, while only 3 were observed in the ERT-SYN006. So as previously discussed in the background at the beginning of this presentation, oxygenes are a type of carbapenemase gene that confer extreme resistance within bacterial organisms. The three oxytypes that were observed within the ERT-SYN006 resistomes were OXA32, 34, and 60. However, as stated before, this is significantly lower than the 12 oxygenes that were detected after ERT exposure only. It is important to note that other resistance genes were altered by the exposure of this antibiotic, including multi-drug efflux transport systems, and they were generally more affected by the ERT resistomes than the ERT-SYN006 resistomes. Increased frequencies of components of the multi-drug efflux transporter systems of aminoglycosan, macrolides, and tetracycline resistance genes were observed in ERT, but attenuated in the presence of SYN, suggesting that SYN006 has the ability to reduce antibiotic resistance, even in genes that are not related to carbapenems. Please note, though, however, that several multi-drug components were still lost between the ERT and ERT-SYN cohorts, although reduction in frequency was more with the ERT only. So the SYN worked well, however, it was not perfect. And that is the conclusion of the results. So now I want to discuss the strengths and weaknesses of this experiment. I think a major strength of this experiment was using the pig model due to, as our discussion beforehand, about how similar it is with the human microbiome. I think that was a huge asset. I also think that the specimen results were consistent between cohorts. Although only 16 pigs were tested, all cohorts except one in observation 2 were all uniform. In addition, the varied statistical analysis using the principal coordination analysis, the Shannon analysis, the Shannon alpha analysis diversity, I think these were very strategically used in order to show different analysis models. In terms of weaknesses, there were only 16 pigs used, which is a very small sample size. In addition, only one carbapenem antibiotic was used, the ERT, and there are many more. So I would like to see maybe a future experiment that expands the panel of antibiotics tested. And in addition, in terms of the antibiotic resistance that was noted in the oxytype, it is very difficult in identifying carbapenemase identification due to the machine needed to be used. The 5 carbapenemase genes that we discussed earlier, KPC, VIM, NDM, OXA, and IM, can be detected on a CARBA-R. However, in order to expand to the other oxytypes, you need a test called the Streck test, which has an expansion of the oxytypes. But I would like to add that these are still new technology models that are being used. So it will be interesting to see how within the future this expands. In terms of future directions of where I would like to see this research going, I think the paper did a very good job at discussing improvements that could be used. So SYN delivery improvement is one way that the research could go to increase by increasing intestinal concentrations. So essentially, you would just adjust the dose of SYN006 to increase the intestinal concentration. And this is shown to I'm sorry. This does not affect ERT serum levels, suggesting that it's not being absorbed systemically. So increasing the dosage will likely improve microbiome protection without having negative consequences. Another modification that could be used is additional zinc. And the reason that this is useful is because the P2A carbapenemase requires zinc for activation because it's a metallo-beta-lactamase. So additional zinc included in this SYN006 formulation, or just given as a supplement to patients, could help with raising the pH of the intestinal fluid so that it could be better delivered and absorbed by patients. So in conclusion, does SYN006 have the potential to protect the gut microbiome and reduce the emergence of carbapenem resistance organisms? I think through this presentation, we have discovered that yes, there is proof that there is protection of the gut microbiome with seeing reduction in ERT only of bacterial strains, while ERT SYN006 is able to maintain these, and potentially reducing the emergence of carbapenem resistance organisms by safekeeping our resistomes. So now some questions to consider. Do you think that SYN006 has the potential for clinical use like SYN004? What are the potential downfalls for using SYN006? Are there any? Do you think advancements like SYN006 are enough to stop carbapenem resistance, as well as antibiotic resistance in general? Consider this scenario. As a healthcare provider prescribing antibiotics, how would you explain to a patient the necessity behind also being prescribed SYN006? And finally, what weaknesses did you observe within this research? And what would you do to fix it? Thank you guys for listening to my presentation, and here are some of my references. Thank you so much.